Okay, so today is our final lab, and what we'll be talking about today is the brain and some of the special senses, the eyes and the ears. Before we do that, I'd like to give you a general layout of next week's exam. There are 50 questions, just like there have been on all other exams. And the difference to this exam is instead of 50 different pictures or 50 different models, there will be 25. And there will be 2 questions related to each model that you see. So you'll have to look at the model, and there'll be always an A and a B that goes along with it. And that's pretty much it. And still the same concept where you'll type in your answer. The exam starts at exactly the time that your lab starts. So if you happen to be starting at 11:30, then you'll start your exam precisely at 11:30, and then it shuts down, I believe, 55 minutes right after that. If your exam happens to start at 2:30, it starts at exactly 2:30 and will log off at exactly 55 minutes after that. Now, you want to make sure that you pace yourself. I've been looking at some of the stats on some of the exams, and exam questions should probably take you 45 seconds typically to answer when you know it. When we start to see students are spending 5 to 6 minutes on a particular question, that's typically not because students are thinking. It's usually because you're using other references and you're looking things up and researching things. So please be careful of that. Those stats are being looked at. And when we start to see 6 minutes on an exam question, it makes everyone a little bit curious as to why. So please be careful with that. In terms of percentages of what type of models and pictures should you expect to see, there's going to be the neuron. There's going to be the brain. There's going to be the spinal cord. You're going to see ventricles. There's going to be flat Stanley for the nerves. There's going to be the reflex arc. There's going to be the eye that we will cover today, the ear that we'll cover today. And then there's the torso model that we went over last week. And the torso model does cover the spinal cord, but in a different view, right? Last week, we looked at the spinal cord from a cross-section view, where we saw the white matter, the gray matter, the spinal cord, etc. And then we looked at the torso from the back, and we saw where the spinal cord ended. We saw the chordae equina, the filum terminale, etc. In terms of the neuron, there'll be about 4 questions on the neuron. There's going to be about 14 questions on the brain model. There will be about 8 questions on the spinal cord, the cross-section of the spinal cord. There'll be at least 4 questions on the ventricles, the different chambers of the brain. There will be about 8 questions on the nerves from flat Stanley. There'll be about 4 questions on the reflex arc, about 2 on the eye, 4 on the ear, and about 2 on the spinal cord from the torso. And when we talk about the brain and the brain model, that includes not just different centers and lobes of the brain, but also cranial nerves. You will have to identify some of the cranial nerves as well. Okay. Now, I also want to make students aware that once you sit and take a final exam, you can no longer withdraw from the class. So I want to make that clear. I'm looking at some of the grades from some students, and I'm a little bit concerned as to why some students are still holding on. Maybe it's financial aid. I get it. If it's for that and you can't withdraw because then you will fall under the credit load and that hurts you financially, I understand. But an F on a GPA is really hard to recover from, and it hurts. So a withdrawal and a W is much, much more forgiving on your transcripts. So please keep that in mind. Okay, so that being said, let's start by looking at the anatomy of the brain. This is that PDF. It looks like it's 37 slides. So let's take a look. For some of you, this will probably be a little bit of review because I know you've covered it already in your lecture. So when we look at the brain from this view, it's a bird's eye view looking down on the brain. We can see that we have these mountains and then we have the valleys. So the mountains, the high spots, are referred to as the gyrus. And then the valleys, these depressions in between the mountains, those are the sulcus. So we have gyrus and sulcus. When we look at the brain from this perspective, we do have two hemispheres. There's a left cerebral hemisphere and a right cerebral hemisphere. And remember, we're looking at this in anatomical position. So this is their right side, this is the person's left, and it's cerebral, not cerebellar. They're different. Cerebral refers to the cerebrum. Cerebellar refers to the cerebellum. Different parts of the brain. When we look at the brain, just like we did the spinal cord, what was interesting was when we looked at the spinal cord, the letter H of the spinal cord, the butterfly, that was gray matter, and then white matter surrounded that. On the brain, it's the opposite, where on the outer part of the brain, the cerebral cortex is gray matter, and the inner part of the brain is white matter. So it's the exact opposite. The brain and spinal cord, it does this flip-flop. In the spinal cord, gray matter's in the middle, but in the brain, gray matter's on the outside. In the spinal cord, white matter is on the outside, but in the brain, white matter is more, it's deeper, it's on the inside. Again, you could see the same concept here, where this is a different view of the brain. This is the left hemisphere, and this is the right hemisphere. These are the right and left cerebellums, but you can see the white matter deeper. And in the periphery surrounding that is the cerebral cortex, which is gray matter. Cerebral is here, cerebellar is here because this is the cerebellum. Now, when we look at the brain, we do have fissures, which are clear delineations that separate certain parts of the brain from one another. So here, going right down the sagittal suture, remember where the sagittal suture was, and when we did osteology, there is a longitudinal fissure. And the longitudinal fissure separates the left hemisphere from the right hemisphere. And we know from lecture that there are different functions of the left hemisphere compared to the right hemisphere. The left hemisphere is more linear, it's more language-based, it's your speech center, it's science, it's math, it's linear thinking. Whereas the right side is more artistic, creative, art, music, facial recognition, understanding the tone in which someone may speak. So right and left side are very, very different, and having healthy left and right parts of the brain that communicate with one another, and they do communicate with one another. Are these white matter tracks that go back and forth called the corpus callosum that allow left and right hemisphere to communicate with one another. Okay, on this part here, we can see the blue venous type of structures. These are important, and it's important to put these in your database because these will be connected to what is discussed in Anatomy and Physiology 2 or AHS 132 when we cover the cardiovascular system. We'll go over the arteries and the veins of the entire body, including blood vessels that bring oxygen to the brain and blood vessels that bring deoxygenated blood back to the heart to become oxygenated. So the main structures that you should be familiar with is the superior sagittal sinus right here, the inferior sagittal sinus, right? It looks like a double rainbow pretty much. So the superior part of the rainbow is the superior sagittal sinus, and then the lower one is the inferior sagittal sinus. And then we have a straight sinus here and a transverse sinus here. And these structures, these blood vessels, will drain deoxygenated blood eventually into the jugular vein. And the jugular vein will find a way of bringing that blood back into the heart. The heart will find a way of pumping that deoxygenated blood to the lungs to pick up oxygen. That's the basic concept there. So we did this longitudinal fissure, which is this way, but then we have a transverse fissure, which will separate the occipital lobe. Back here is the back of the brain. This is the occipital lobe, which is your visual center. And this is your cerebellum. So between the occipital lobe of the cerebrum and the cerebellum is a transverse fissure. The tentorium cerebelli is this membranous soft tissue structure here, which is the soft tissue structure that will separate not just the fissure, but it is the membrane that you can find here that separates the cerebellum from the occipital lobe. Okay, the fissure is the gap or the space, but the tentorium cerebelli is the membrane. Now, you'll notice that there is this red strip here, and then this blue strip here. And in between the red and the blue strip is a central sulcus. It's a very, very important landmark, this central sulcus. This is the frontal lobe. Back here is the occipital lobe. So this is anterior, back here is posterior. So the red structure, which is in front of the central sulcus or comes before the central sulcus, is referred to as the pre-central gyrus. Pre-central gyrus. And what's behind it in the blue is the post-central gyrus. So you have pre in the front, pre-central gyrus, and post-central gyrus in the back. And those two are separated by this central sulcus. Functionally speaking, the pre-central gyrus is where all of your motor impulses start. And the post-central gyrus is where all of your sensory input ends up. So in order for you to create a muscle contraction, those impulses need to start somewhere. that thought of moving a part of your body is generated here in the precentral gyrus. But where all of your sensory input ends up is your post-central gyrus. So can you tell that you have pain in your shoulder? How can you tell that you have pain in your left knee compared to your right knee? Even though you feel the pain in these particular parts of the body, remember the parts of the body only contain receptors. The receptor picks up the signal and sends it eventually up here to the postcentral gyrus. And if you were to look in your textbooks and your lab books and you look at the term homunculus, you will see that the homunculus is the depiction of the entire body stretched over this part and this part of the brain, that each particular portion relates to a specific part of the body. And it is the brain that picks up the pain. Pain is only felt in the brain. Okay. So now this view with the central sulcus and the pre and post-central gyrus, looking at it from the side view, here's the lateral view, we can see it here. So here's the central sulcus once again, and it is this depression that starts at the apex, like right up here at the top of the brain, and this sulcus goes all the way down. It makes it all the way through to this temporal lobe, and then it stops. So that's your central sulcus. Here's your frontal lobe. The green back here is your occipital lobe, where the occiput is. So this again is your pre-central gyrus, which is called the motor strip. And this is your post-central gyrus, known as the sensory strip, or somatosensory, somatomotor. Somatosensory, somatomotor. This pre-central gyrus is located and is part of the frontal lobe of the brain, whereas the post-central gyrus is part of the parietal lobe of the brain. Parietal lobe, this is part of the frontal lobe. Now there's a lateral sulcus, which is this, that separates this portion of the frontal lobe, this portion of the parietal lobe, this portion of the occipital lobe. It separates it from the temporal lobe. This is the temporal lobe. So this is your lateral sulcus here. Now this view is an inferior view looking underneath the brain. Here is your medulla. Here is your pons. And up higher, here is your midbrain. And we can see this long nerve right here is the olfactory bulb. The tip is the olfactory bulb. This is the olfactory nerve or the olfactory tract. This is picking up the sense of smell. This nerve is the one that travels through the cribriform plate. That was why the cribriform plate, or the sella turcica, was such an important landmark when we did the osteology and we did the skull, because these are going to pierce right through the olfactory bulb. The olfactory bulb is going to pierce and have these extensions right through the cribriform plate and the sella turcica. Then we have the optic nerve, which is cranial nerve 2. This is cranial nerve number 1. This is cranial nerve number 2. There's a right and a left optic nerve, and where they crisscross in the center is referred to as the optic chiasm. The optic chiasm is where both right and left optic nerves, cranial nerve 2, crisscross. And right here, that gland is the pineal gland, or the pineal gland. Sorry, the pineal gland, yes. And when you have, sorry, not the pineal gland, having a junior moment, pituitary gland. When the pituitary gland enlarges, right, if someone has a pituitary gland tumor and it enlarges, one of the symptoms that people have that have pituitary gland tumors is there are these visual deficiencies or impairments or deficits, right? The eyes just start to dysfunction depending on where that tumor is pushing on the optic nerve. Okay, so there are these visual issues. So you have the olfactory bulb, olfactory nerve, optic nerve, optic chiasm, which is where both crisscross. Now we can see the different lobes of the brain labeled here. Here is the frontal lobe, and again, the precentral gyrus is part of that frontal lobe. This is the parietal lobe, and the postcentral gyrus is part of the parietal lobe. All the way in the back, occipital lobe, which is your visual center, and temporal lobe here, which is for hearing and even taste. Your frontal lobe deals with your, this is kind of like your personality. A lot of your personality comes from the frontal lobe, your drive, your ambition or lack of, your drive or lack of. Many OCDs and obsessive compulsive disorders start up here in this portion of the frontal lobe. There are impulsivity, where people don't know how to hold on to their words, where they just come out and then they go, I shouldn't have said that, right? The ability to inhibit and not say certain things are controlled by the frontal lobe. When little Johnny pushes his little brother or sister down the stairs, and the mother or father or grandparent or family member says, you know, little Johnny, you really shouldn't push your brother or sister down the stairs, they can get hurt. Okay. And then the next day they do it again. Why did you push your brother or sister down the stairs? I don't know, right? Even though they know that there would be a consequence for their action and someone could get hurt, the inhibitory center, the ability to your brain to speak to you and connect and say, I shouldn't do that, there is a consequence for that action, the dots just don't connect. The frontal lobe inhibits activities like it slows things down. If you think of a baby's heart rate is beating very, very quickly. A baby is breathing very, very fast. And it's only until the age of 4, 3.5, 4, even 5, when the frontal lobe really starts to mature, that now the frontal lobe, the inhibitory center, can start to slow down and inhibit that fast heart rate, and it slows it down. It can inhibit or slow down the fast breathing pattern and slow it down. Frontal lobe, to inhibit when you need to urinate and go to the bathroom, there's a signal that says, I have to go to the bathroom, and you run to the bathroom. Babies wear diapers for a reason. They can't inhibit. That frontal lobe is not yet developed. Okay. Parietal lobe is all about your perception and awareness of things. Doc, for some reason, why does my left ankle look bigger and more swollen than my right ankle? And I would tell them it's not. They are the same size. No, 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 doc, I swear, look at it. Look at my left ankle. Look at my left wrist. It's bigger than the right half of my body. It's so much puffier and larger. Don't you see it? No, I'm sorry, I don't see it. It looks the same. But there's something in that individual that's going on in their perception center that perceives that one side of the body is larger than the other. So then we could take a tape measure and objectively measure the ankle, or objectively measure the wrist, and compare both sides and show that individual that they are, in terms of measurement, the exact same size. They can see that, they can read it, but then it doesn't connect, and they say, yes, but look at my wrist, look at my ankle. The left one is still bigger than the right. I don't care what those numbers show. Okay, this is a distorted perception based on the wiring within the brain. The occipital lobe back here is your visual center. There are certain parts that allow you, even though the eyes, again, the eyes are the receptor for vision, they have the receptors for what you see. Those signals are eventually sent all the way back to the brain, and your brain does the seeing and a lot of the interpretation of what you see. All right, so here again, this is your central sulcus, and then there's your pre-central gyrus. Back here in the blue is your post-central gyrus. There's your frontal lobe. Post-central gyrus, which is this primary sensory area. Again, here's your temporal lobe, occipital lobe, frontal lobe. This is your parietal lobe. Post-central gyrus is part of the parietal lobe. Pre-central gyrus, part of the frontal lobe. Here's the back. This is the occipital lobe, and we said this is the visual cortex. This is the part of the brain that allows you to see objects, but then it sends a signal to the visual association area that helps you interpret what it is you're seeing. So if you were to see an actor or an actress somewhere in the public, your eyes could actually see him or her, and then it will send that message to the visual association area, and you start to process and go, where do I know him or her from? What movie was he or she in? Right? So they connect and they communicate with one another. But if someone slips or falls and they hit the back of their head, they can go blind because this is the part of the brain that actually does the seeing. The eyes only have the receptors for vision. Here is your temporal lobe, which is your hearing center. This is your auditory cortex. It allows you to hear, and then it will relay it to this part, which is the auditory association area. So you can interpret and go, oh, I know who sings that song. Okay. When we separate the temporal lobe from the rest of the brain, we can see this orange structure in here, known as the insula. Inside is the insula. It's the orange. When we separate the brain into the two hemispheres, what we're looking at down here, this white structure from here down, this is your brain stem. And your brain stem is made up of three parts of the brain: your medulla, your pons, and this section here, the midbrain. Medulla, pons, and the midbrain. This is the cerebrum, all up here. These are the different parts of the cerebrum, like frontal lobe, parietal lobe, occipital lobe. In the center here, here's the corpus callosum. This is that white band that I said helps the left and right side of the brain to communicate with one another. The septum pellucidum is this clear membranous structure that separates the ventricles, the lateral ventricle 1 from lateral ventricle 2. It's this clear sheath, this membrane called the septum, like a wall, like a nasal septum, septum pellucidum. This fornix right here, fornix, this is part of the emotional brain. Fornix. Again, corpus callosum. Here is the separation of the lateral ventricles 1 and 2 by the septum pellucidum. And then here is the fornix. When we look at this structure right here, this round almond-like structure, this is the thalamus. And the thalamus is the relay station for all of your sensory input. So all of the sensory input comes up the medulla, through the pons, into the midbrain, and will make a relay here at the thalamus, and the thalamus will end up shooting it up to the post-central gyrus, which is where all the sensory input finally ends up. But it is the thalamus that is the relay station. The yellow circle that looks like the sun, that's right there, that's the interthalamic adhesion. The interthalamic adhesion. Thalamus, interthalamic adhesion. Again, thalamus, interthalamic adhesion. Now, under the thalamus, right, this is the thalamus, inferior to it here, this is the hypothalamus. Now, the hypothalamus is the major control center of the ANS, meaning the autonomic neural system. When you think of the autonomic neural system, the self-regulating, self-law, you should be thinking about the sympathetics and the parasympathetics. Okay, so the hypothalamus is that control center for the autonomic neural system. And when you get to AMP2, we will discuss the connections between the hypothalamus, the pituitary, and so many other parts of the body, because the neural system and the endocrine system is what regulates and controls about every function of the human body. The neural system does it through electrical impulses and messages, whereas the endocrine system uses hormones as chemical messengers. Okay. Hypothalamus, thalamus, hypothalamus. Again, different view, but here is the thalamus, interthalamic adhesion. Now, you heard me talk about the hypothalamus and that there are connections to the hypothalamus to the pituitary gland. Now, we don't want you to confuse pituitary gland with the pineal gland as I did earlier, right? Pituitary is here, pineal gland is back here. The connection or the stalk between the hypothalamus and the pituitary is called the infundibulum. So thalamus, hypothalamus, infundibulum, and pituitary gland. The pituitary gland sits in this cavity, this fossa, called the hypophyseal fossa, aka sella turcica, the Turkish saddle that you learned when we did osteology. We wanted you to know that because it is an important structure. It is where the pituitary gland sits in. Okay, we're going to get into the ventricles soon, which is where you'll hear the term choroid plexus. The choroid plexus is part of all of the ventricles, and they are responsible for producing and recycling the cerebral spinal fluid, the CSF. So here is the choroid plexus of the lateral ventricles. It's a vascular system right here. Just inferior to that, here's the pineal gland. And the pineal gland is in the brain, but it has endocrine function, and it is responsible for secreting melatonin. And melatonin is secreted at night, and it helps to tone you down. Melatonin. It's not melanin, right? Melanin is for skin pigmentation. Melatonin helps you stay calm. Here again is the choroid plexus, a little closer view. There's the corpus callosum here. Septum pellucidum. Thalamus. Hypothalamus. Interthalamic adhesion. Pineal gland. The brain stem, here is the brain stem, and the three parts of the brain stem are the medulla, from inferior to superior, medulla, pons, and the midbrain. The midbrain here, when we rotate it around, we're going to see these two bumps, but there's two bumps on the right side of the brain, and then there's another two bumps on the left side, right? Don't forget, this is only half the brain. So the four bumps combined, two on the right and two on the left, are part of the midbrain, and it makes up what's called the Corpora Quadrigemina. The Corpora Quadrigemina literally translates to the body of four. There are two on top and two on the bottom. The two on the top are called superior colliculi, and the two on the bottom, inferior colliculi. Superior colliculi, inferior colliculi. All four combined, Corpora Quadrigemina. Medulla, pons, and this section here, the midbrain. So here is now what we're doing is we're taking this section here, right and left side of the brain, and we're going to rotate it so that we're looking at the back side. So here, what we were looking at were the superior colliculi and inferior colliculi of just half, but now as we rotate it, we can see all four. 1, 2, 3, 4. So it's called the Corpora Quadrigemina. Quad is four, body of four. Two of the superior colliculi that deal with visual stimulus, and then the inferior colliculi is one of the relay centers for your auditory or hearing information. So you got your medulla, your pons, and the midbrain. And the Corpora Quadrigemina is part of the midbrain, and there's two superior colliculi and two inferior colliculi. Here's your cerebellum. When we open up and dissect the cerebellum, there's the center part that connects the two. That's called the vermis, means worm. This is the right cerebellar hemisphere, left cerebellar hemisphere, right? It's not left cerebral hemisphere. Cerebral refers to the cerebrum. Cerebellar refers to the cerebellum. When we open that up, we can see it kind of looks like a leaf, doesn't it? It looks like the structures that you can see when you look at a leaf that has fallen from a tree. The inner part, which is white matter, is called the arbor vitae. It's the veins of the leaf. It literally means tree of life, arbor vitae. The gray matter is called the cerebral cortex, also known as folia, like foliage, F-O-L-I-A, folia or foliage. That is gray matter. Now, we're going to look at a structure. Let me just scroll up here for a second. Here, this part of the brain is called the diencephalon. All of these structures here are part of the diencephalon. And within the diencephalon, in this entire section here, we're going to see the ventricles, the four ventricles. So that's what we're looking at here, are the ventricular system. So we're looking at it from back to front here. There's two lateral ventricles. This is a lateral ventricle, and this is a lateral ventricle, sometimes called lateral ventricle 1 or lateral ventricle 2. There's no clear delineation whether the right one is number 1, the left one is number 2. So it's just lateral ventricle 1 and lateral ventricle 2. There are four altogether. So again, lateral ventricles 1 and 2. The fluid, the cerebral spinal fluid that's here, is going to exit and drain and go down in this direction. So it's going to go up and then come down. In order to go down into the third and fourth ventricle, it's got to drain through the interventricular foramen of Monroe. So this is the interventricular foramen, also known as the interventricular foramen of Monroe. That's how the CSF goes from the lateral ventricles, 1 and 2, into the third ventricle. It's got to go through the interventricular foramen of Monroe first. So now you've got lateral ventricles 1 and 2. We're rotating the ventricles a little bit. Here's the third ventricle. And the interventricular foramen of Monroe is blocked by this lateral ventricle, but it would be up in here. Then it drains into the third ventricle. Then the cerebral spinal fluid has to go from the third ventricle into the fourth ventricle. And the way it gets from the third into the fourth is through the cerebral aqueduct of Sylvia, or cerebral aqueduct of Sylvis. That's this chamber here. And then after it's in the fourth ventricle, it can exit through the lateral aperture on both sides here, or the median aperture. And the median aperture is going to drain into the central canal. So now you have to think of the cross section of the spinal cord. And if you think back to the cross section of the spinal cord, in the gray matter, the letter H, or the butterfly, where the gray commissure is, right in the center was a little pinhole. And that's where we said cerebral spinal fluid flows. So the median aperture is going to lead the CSF, or cerebral spinal fluid, down the central canal. Okay, so that brings an end to the brain. Now let's take a look at, let's see, I think this is a little bit more of the brain and a little bit of the special senses. So on this cross section here, this is the thalamus, interthalamic adhesion, hypothalamus, medulla, pons, midbrain. And this is where we have the superior and inferior colliculi called the Corpora Quadrigemina. Back here is the cerebellum. This is the arbor vitae, the white matter, the tree of life. And around it is the folia, like foliage, leaves. It's the gray matter. Here is the thalamus, hypothalamus, infundibulum, pituitary gland, pineal gland. Okay, hypothalamus is here. Corpora quadrigemina is here, made up of the superior and inferior colliculus. Okay, let's see if there's anything new here. These are all structures we have spoken about, with the exception of one from this view. We did the corpus callosum. We talked about the fornix. Here is the vascular choroid plexus that surrounds the lateral ventricles. There's your corpora quadrigemina, part of the midbrain, pons, medulla, cerebellum. Here's the pituitary gland. And I showed you from the other view, when we looked underneath the brain, there was the optic nerve. And where the optic nerve crisscrossed, that was called the optic chiasm, the optic chiasm. So this is the optic chiasm from the lateral view. And this is why when the pituitary gland increases in size and pushes up against the optic chiasm, it can affect your vision because it carries visual information to the brain. Interthalamic adhesion, thalamus, hypothalamus, pituitary gland, optic chiasm, pineal gland. If I don't mention it, then don't worry about it. If you don't see me or hear me mention it, don't worry about the structure. From this view, corpus callosum, the white structure here. On this view, choroid plexus, interthalamic adhesion. This whole structure here is the thalamus. Underneath it, hypothalamus, infundibulum, leading to the pituitary gland. This is the optic nerve and the optic chiasm. Medulla, pons, midbrain, superior, inferior colliculus, superior, inferior colliculus, cerebellum. The veins are the arbor vitae, white matter. Folia is the gray matter. Okay, from this structure here, superior colliculus, inferior colliculus. Right here is where the fourth ventricle is. And you can see it's just anterior to the cerebellum. Here's the cerebellum, there's the chamber, the fourth ventricle. Medulla, pons, midbrain. 26 is the choroid plexus. 22, interthalamic adhesion. Thalamus, hypothalamus. Cranial nerve number 1. Here's the olfactory bulb. Here's the olfactory tract or olfactory nerve for sense of smell. Optic nerve is cranial nerve 2, where they crisscross, optic chiasm. Here's your pituitary gland. From this view underneath, again, here's the olfactory nerve, cranial nerve number 1, and here's the optic nerve, cranial nerve number 2, where they crisscross, optic chiasm. Pituitary gland. You should know for lecture and lab the 12 cranial nerves. You should know them by their name and by Roman numeral. I won't ask you sensory or motor in terms of their function. Maybe that's something you need to know for your lab, for your lecture, but not for lab. But you do need to know the names of the 12 cranial nerves and what their primary functions are. Right? Olfactory nerve is for sense of smell. Optic nerve for vision. Cranial nerve 3, cranial nerve 4, and cranial nerve 6, they all move the eye. The oculomotor nerve, the trochlear nerve, and the abducens nerve go to those 6 muscles of the eye. We covered them when we did muscles. They were the medial rectus, superior rectus, lateral rectus, inferior rectus, and superior and inferior obliques. Those are controlled by cranial nerves 3, 4, and 6. Cranial nerve 5, the trigeminal nerve, deals with sensation to your face and the muscles of mastication, which means the chewing muscles. The temporalis, the internal pterygoid, the masseter, and the external pterygoid. Those are innervated by cranial nerve number 5, trigeminal nerve. Cranial nerve 7, facial nerve, deals with the muscles of facial expression, like smiling, raising your eyebrows. It also deals with the sense of sweet, sour, and salty to your tongue. So sweet, sour, salty 7, and it deals with facial recognition, facial expression, the muscles of facial expression. Cranial nerve 8, vestibulocochlear nerve. When you look at this, it's vestibular and cochlear. When we look at the ear, I will show you the vestibule and I'll show you the cochlear. The cochlear deals with the hearing. The vestibular portion deals with your sense of balance. Glossopharyngeal deals with the muscles of the back of the throat. It deals with swallowing. It deals with the gag reflex. That's cranial nerve 9. Cranial nerve 10, the vagal nerve. Vagal nerve controls pretty much all of your internal organs. There's innervation to the stomach, to the heart, to the lungs, to the intestines, to the liver. Every organ basically gets nerve supply from the vagal nerve. It makes up about 90% of your parasympathetics. Your parasympathetics are cranial nerve 3, 7, 9, and 10, and sacral segments S2, 3, and 4. That's the parasympathetic. Cranial nerve 11 is known as the spinal accessory nerve, and that controls the upper trapezius muscle and the SCM, sternocleidomastoid. And then cranial nerve number 12 is the hypoglossal, that moves the tongue around. Okay, let's look at the eye and look at some of the major parts of the eye that you should be familiar with. The black central part is known as the pupil. The colored portion, some people have blue eyes, some people have brown eyes, some people have green eyes, that is the iris. The sclera is the white portion out here. The cornea, I'll show you from a different view. Your tear ducts, your tear glands are up here. That's the lacrimal gland. It's superior and lateral. And then your tears roll down inferior and medially. So it starts superior and lateral, and then it cleans the eyes. It drains into the medial portion, into the canal, and then into the nose. And then these turbinates swell up because the tears are pretty dirty. So that's why you get all stuffed nose when you cry, or you get teary. So when we look at the eye, the colored portion is the iris. The dark circle, that's the pupil. That's what can constrict or dilate, depending on light. When light shines in, your pupil constricts. In the dark, for better vision, your pupil opens up and dilates. The clear translucent membrane in the front is the cornea. And the cornea is somewhat continuous with the white part, which is the sclera. So the cornea is translucent, it's clear. The cornea is the white part. And you can see the muscles attached. The superior rectus, lateral rectus, inferior oblique, inferior rectus muscle, right? These are the muscles that are controlled by cranial nerve 3, 4, and 6. When we look at the different portions of the eye, again, here's the cornea, which is the clear part, and we can see that the cornea is continuous with the white part, which is the sclera. And then when we take off the top layer, we can look at the different depths, right? The different portions of the eye from anterior to posterior. So here's the cornea, and you can see here is the lens, and this anterior chamber right here is the anterior compartment, and there's a type of fluid in here called aqueous humor. Here is the lens, and behind the lens is the posterior compartment, which is filled with vitreous humor. Here is your pupil. Here is the iris. You can see the sclera. Here is the ciliary muscle that helps make the eye constrict. Here is the vascular layer called the choroid. That's the brown layer in here. This is the choroid. So the outer layer is the sclera. As we go in a layer, the middle layer, that's your choroid. And then when we go way, way deep in the inside, right in here, that's where we're going to find the retina, the innermost layer. Here's the lens from this view, from the inside. And then you could see the ciliary muscles to help make the eye constrict. From this perspective, we have the pupil, the iris, the ciliary muscles, the white part, is the sclera. The cornea is that clear part that is removed from here, so we don't see that. So the sclera is the outermost layer, the middle layer is the choroid. That's the vascular layer. Again, you can see the sclera is the outer layer. Then we can see the choroid is the middle layer. And then all the way on the innermost portion, the inside of the eye, that's the retina. Here's your iris, the colored portion. This would be the pupil, half of the pupil. And the lens is deep to that or posterior to that. And then the posterior chamber, that's where you have the vitreous humor. Here is where the pupil would be. This is where your lens would be held. This is the indentation, right, or the fossilized portion of where the lens sits. And then this roughened portion is where the ciliary muscle attaches to it and changes the shape of the lens. Then on the innermost part, this is, let's go back here. The middle layer is the choroid, and then the deepest part, the inner part, is the retina. And then the optic disc is right here at the back of the eye. That's where you have the entrance of the nerve and the blood vessels, the arteries and veins. And again, different view from anterior to posterior. So here is the cornea. We said that the cornea is continuous with the sclera, which would be here. The middle layer is the choroid. And then the innermost layer is the retina. Here's the pupil. Here's the lens. And the lens is held into the ciliary muscle. You can see there are these ligaments that attach to it. So when the muscle constricts, it pulls and creates different size and shape of the lens. It can alter it to change the vision. Back here is the optic disc, where you have the optic nerve and you have the artery and vein. And again, another view just showing all the same structures from the cornea to the iris, the colored portion, to the light going through the pupil, through the lens. You can see the suspensory ligaments here. You can see the three layers. You can see the sclera. You can see the choroid. And the innermost is the retina. You can see the optic disc in the back, which is where the nerve, the artery, and the vein enter into the eye or exit, whichever way you want to refer to it as. You remember the eye with the muscles, superior rectus, lateral rectus, all the way in the bottom. Can't quite see it too good, would be the inferior rectus. Through the top is the superior oblique, and on the bottom would be the inferior oblique. Pupil, iris, sclera. The cornea is removed. That's the translucent anterior portion. Let's look at the ear. So how do we hear? Sound waves are collected through the funnel, right? The funnel is the pinna or the auricle of the ear. That's the outer part of the funnel. It collects the sound waves. The sound waves go in and bounce up and down in here through the external acoustic meatus. The sound waves come in and hit the tympanic membrane. The tympanic membrane then vibrates. As the sound comes in, the tympanic membrane, like the eardrum, this thing is going to vibrate and send this vibration through the three smallest bones of the body, the malleus, the incus, and the stapes. The three red ones, malleus, incus, and stapes. So when these vibrate, it will put this tugging force on this oval window here, and this suction of movement back and forth from all of this vibration will send a vibratory impulse here into the inner part. Looks like a snail, That snail is called the cochlea. Remember I said cranial nerve 8 is called vestibulocochlear. And that's because there are two divisions of this yellow nerve. One is the cochlear portion, the other is the vestibular. The vestibular portion goes to these semicircular canals in which there is fluid in here that as your head moves in different positions, it sends signals so that you don't fall over and you can maintain your posture. The cochlear has inside something called an organ of Corti, which is the auditory receptor for sound. So your body is taking this vibratory, it's taking vibration, a vibrational frequency, sending it into the cochlear portion of the vestibulocochlear nerve, and then it's going to send it into the brain where the temporal lobe is. Okay, here are the canals. These are the ones that are involved when like mom or grandma or grandpa turn their head and then everything feels like it's spinny and they get vertigo. This are the semicircular canals when a crystal gets trapped in these tubes. It's going to give them what's called benign paroxysmal positional vertigo. It is fixable. I happen to treat it in my office. It's poorly managed with motion sickness medication, but can typically be fixed in under 5 minutes of time. The only ones that can't be fixed is if they are chemically induced. People that are on certain blood pressure medications, they're ototoxic and they can throw off a person's balance and can also give them the sense of tinnitus or ringing in the ear. Okay, here is another view of the same structure. Here is the tympanic membrane, malleus, incus, and stapes. Now we get that vibration that's going to move through the cochlear. As it moves through the cochlear, that vibratory sense is now translated into the cochlear portion of the vestibulocochlear nerve, cranial nerve 8, the V with the 3 ones. Here are the semicircular canals. And there's 3 of them. There's an anterior canal, a posterior canal, and a lateral canal. So when your head goes forward, you're stimulating the anterior canal. When your head extends back, you're stimulating the posterior canal. And when you turn your head left and right, it's stimulating the horizontal or transverse canal. And there are these little crystals that move and stimulate these little hair-like structures in each of these canals. And it sends signals to your eyes, it sends signals to your neck muscles, and it sends signals to your cerebellum, which controls your balance and coordination. This is why when people have vertigo, their sense of balance is completely thrown off. When we look at the ear model, again, here is the pinna, or the auricle of the ear. Here's the external auditory meatus. Here's the tympanic membrane. Then you have your malleus, your incus, and your stapes. Here are the semicircular canals. The semicircular canals attach to the vestibule. And on the other side is the cochlear for hearing. Let's turn it around and get a better view of it. So here is the external auditory meatus, so sound waves are coming in through here. It hits the tympanic membrane and then hits the malleus, the incus, and the stapes. Stapes means stirrup, like a horse stirrup, putting your feet, if you're on a horse, through the stirrup. And then it connects to the oval window. And then that vibration comes through here into the cochlear, through the cochlear portion of the vestibulocochlear nerve. These are the semicircular canals. This is the anterior canal, posterior canal, horizontal canal. All 3 of these semicircular canals, which contain fluid, attached to the vestibule. When these crystals stay lodged in the canals, you get vertigo. They're supposed to drop and fall back into the vestibule. When the crystals or stones stay lodged in here, the person gets the feeling that they're still moving when they're not. And that's what makes them feel sick, and they feels like they're spinning, and the eyes go into this rapid firing back and forth called nystagmus, and they end up vomiting from it. When you take everything else out and you just show the structures, here's the tympanic membrane, the malleus, the incus, the stapes, oval window, the vestibule with the anterior canal, posterior canal, and the lateral canal. The snail structure is called a cochlea. Here's the cochlear nerve and the vestibular nerve combined together. It's the vestibulocochlear nerve, cranial nerve 8. The rest of these you can review on your own because we did do these last week. This is just a good slide because here it points to the numbers, right? The numbers have arrows, and then these numbers go hand in hand. So you can self-test yourself, as you can self-test yourself here as well with the structures on top and the name of the structures down below. These are all review. We did the neuron. 3 main parts of the neuron are the dendrites. This is the axon. In the center is the cell body. And these funnel-looking structures are called the axon terminals. This is the axon terminal. What's in these axon terminals are neurotransmitters. And neurotransmitters could either be excitatory, like an on switch, or inhibitory, an off switch. What surrounds the axon is myelin. This is cholesterol or a fatty structure. Okay. And the Schwann cell in the peripheral neural system is what myelinates nerves. The Schwann cell myelinates nerves in the peripheral neural system. You learned that the oligodendrocyte myelinates in the central neural system. The invagination between this Schwann cell and this Schwann cell is called the node of Ranvier. And the layer of tissue that surrounds that axon and this part of the neuron is called an endoneurium, right here. So you have the main parts of the axon, the cell body, the dendrites, and the axon terminal. What surrounds the axon is the myelin or the myelin sheath. This is the nucleus of the Schwann cell, which is laying down the myelin to insulate and protect the axons. We did this last week. We went over the parts of the reflex arc. We said there were the receptors, the sensory fiber or afferent fiber. Here is the interneuron. Here's the efferent fiber or the motor neuron, and then the effector.